Hi, my name's Dr. Jeremy Ray, and today we're going to talk about DNA structure and replication. These are your learning objectives, which you'll find very useful when you're revising this subject. So we're going to start off with the general structure of the human genome. We'll talk about function and relative proportions of 5 important types of DNA sequences, including centromeric DNA, repetitive DNA, telomeres, the transcribed region, and regulatory DNA. We're going to describe the concept of transposable DNA and its role in genome diversity and formation of pseudogenes. We'll describe the basic structure of DNA, and it's important to be able to differentiate this from RNA. We'll review the structure of nucleotides and the roles of the base 3' hydroxyl group and 5' phosphate group. Explain the functions of the different DNA polymerases. So we've got alpha, we've got delta, and we've got epsilon, for example. Then we've got helicase, beta sliding clamp DNA ligase, and 2 types of toposomerases. These are all involved in DNA replication. We'll explain why synthesis occurs only in the 5' to 3' direction. We'll explain how DNA polymerases carry out proofreading. We'll describe the complex for initiation of replication and its relationship to cell cycle control. And lastly, we'll explain the production and maintenance of telomeres. Okay, so the Human Genome Project that began in the mid-1980s and was completed in 2006. And our genome is 3 billion base pairs long. And of course, it's divided into chromosomes. We have 23 different chromosomes. And of these, 22 are the autosomes, which are non-sex linked chromosomes. And then, of course, we have the X and the Y sex chromosomes. And females have 2 X chromosomes and males have an X and a Y chromosome. So what we have here is a graphical representation of the human diploid karyotype, because we, of course, are diploid organisms. We have 2 copies of each of the chromosomes in our cells. And a karyotype is a very useful tool, and it can be performed to see, for example, if someone has genetic abnormalities, such as an extra or missing chromosome. So chromosomes, of course, are made from chromatin. Chromatin is a complex of double-stranded DNA, which is wrapped around histone proteins to form nucleosomes. And then nucleosomes are further organized into solenoid structures, which become supercoiled and make up our chromosomes. This here is a metaphase chromosome. So it's the form of chromosomes that you only see during mitosis, specifically the metaphase stage. So what happens that during cell division, mitosis, is chromatin becomes more dense. It condenses into these chromosome structures, which we can see at metaphase. And the rest of the time, it's actually in a more relaxed state to enable gene expression and so forth. So one of the features of duplicated chromosomes, and we've got one here with 2 sister chromatids, and these are connected together by this region called the centromere. And this is a very AT-rich region. And in humans and other mammals, it's about 1 million base pairs in length. And it's bound by nucleosomes, which contain a histone HV variant protein called CNPA and other centromere binding proteins. And collectively, we call this complex the kinetochore. And this is the anchor point for microtubules that make up the mitotic spindle, so that during the process of mitosis and the segregation or separation out of chromosomes, this is an essential structure. So that this is the point you recall that the chromosomes, they're pulled apart by the mitotic spindle and they go to the opposite poles of the cell. So what we see is there's instability in this region, and this instability that causes alterations and breaks in DNA are actually associated with a number of different human diseases, and that includes numerical abnormalities when you have extra or missing chromosomes, and also this syndrome called immunodeficiency centromeric instability and facial dysmorphism syndrome, ICF. And here's an individual with this syndrome, and that is linked with instability in the centromeric region. So looking at the overall structure of the human genome, and to remember that a small proportion of our genome actually comes from mitochondrial DNA. So the vast bulk is in our nucleus in the cells, and then we have about 33 genes which are in our mitochondrial DNA. Recall mitochondria, the powerhouses of the cell. They produce most of our ATP, and they have mitochondrial DNA, but it's a very small percentage of our overall genetic material. So as mentioned before, human genome is over 3 billion base pairs in length, and it contains roughly 25,000 protein coding genes. So only about 1.5% of your total DNA is actually protein coding genes. So what's the rest of it? Well, let's have a look at this in more detail. So once again, there's your genome, there's your mitochondrial DNA, and we can split it up into what we call genic and related DNA. So that's DNA that's to do with coding genes and all their related sequences to do with coding genes. So the coding regions of genes, of course, are exons. And over here we have the non-coding parts, and that includes, of course, the introns. So you recall that when genes are expressed, they undergo, the RNA gets spliced, and we have to remove the non-coding introns out. And also we have these pseudogenes. And what do we mean by that? Well, what they are is actually remnants of coding genes that have lost their coding function. However, it's believed they still have regulatory functions. And pseudogenes, one of the processes in which they're formed is by mobile genetic elements. So if we look at the other parts of the diagram, and now we'll go over to the right-hand side, this is the extragenic DNA, so the DNA that is outside of coding regions. It's nothing to do with expressed regions of DNA. And we can further divide that up. We have unique sequences, and then we have repetitive DNA, and then we divide that further into tandem repeats and interspersed repeats. So definition of a tandem repeat, it's a sequence of 2 or more DNA bases that are adjacent to each other on a chromosome, and we find them in centromeres, as we mentioned earlier, and also we find them in telomeres. And they are very useful as markers of disease. And we have these 3 types of satellites. So satellite is a large array of tandemly repeating non-coding DNA. And then we have different lengths or sizes of these satellites. We have micro and mini satellites. So the interspersed repeats, which are scattered throughout the genome, excuse me, we have 4 categories. We have long terminal repeats, we have the short and the long interspersed nuclear elements, signs and lines, and these all undergo transposition. They are mobile DNA elements, and then we have the DNA transposons. So we believe that these mobile genetic elements contribute to the formation of the pseudogenes over here. Sometimes we use the term retroposons or retrotransposons to describe this 3 group of mobile genetic elements. So looking at microsatellites in a bit more detail, so they're defined as 2 to 6 base pairs repeated up to 50 times, and they are highly polymorphic. We see a lot of variation between individuals, for example, so they have a range of uses, for example, in DNA fingerprinting, and also as markers of a range of genetic diseases. So if we use the method PCR, polymerase chain reaction, this is a very powerful molecular technique which we use increasingly these days, and you might know that it's the basis of the coronavirus test, is that we can easily screen a large number of family members very quickly for microsatellite polymorphisms or variation in microsatellite sequences. There's a group of disorders that are associated with microsatellite instability, and we call them the trinucleotide repeat disorders. So we see changes in these sequences that can, in some cases, cause disease. An example of a TNR is Fragile X syndrome, which is shown in this diagram here. This is a leading cause of intellectual disability, primarily in males, though it does occur in females as well. And this is associated with an unstable repeat, CGG. And once we get over a certain number of these repeats, then you exhibit the symptoms of Fragile X. And in addition to the intellectual disability, you also have facial abnormalities, as shown here in this diagram. And a couple of examples of trinucleotide repeat disorders is Huntington's disease and myotonic dystrophy. So Huntington's is a progressive loss of cognitive function that usually appears in middle age, and also myotonic dystrophy, which is a muscle wasting disorder. So transposons, they're also called jumping genes, mobile genetic elements. They can move from one part of a chromosome to another, or they can move between different chromosomes, as shown in the diagram. And as a result, they may increase or decrease the amount of DNA in the genome. And you can imagine if this was to occur in gametes or sex cells, then you have a change that is potentially heritable that could be passed to the next generation. Up to 1/3 of the entire genome may be made of repetitive sequences that come from degenerate copies of transposable elements. We classify the transposons by their type and size and how they actually move. So here we have different categories of transposons. We have class 1, also called retrotransposons, and the DNA gets transcribed into RNA, and then they utilize an enzyme called reverse transcriptase. And what that does, it makes a DNA copy of that RNA, and then it's able to insert in a new location somewhere else in the genome. So we have two types of class 1. We have the long interspersed elements or lines, and here's one here. And also what we can see in this diagram is the relative percentage of DNA that these different elements take up. And this one here, for example, is 17%. So it's one of the largest proportions of mobile element. So long interspersed elements, find them in high numbers, and they have the enzyme reverse transcriptase and an RNA polymerase promoter that is contained within the gene, within the element itself. And then we have the short interspersed elements or signs, and they're smaller and they lack the reverse transcriptase enzyme. So they need other mobile elements to move around. And the most common type are called allo sequences. Then we have class 2 or DNA transposons. They consist of only DNA that moves around from locus to locus, and they have a transposase encoding gene present. So clinically, we're interested in them because they act as mechanisms for spontaneous generation of mutations. This is called insertional mutagenesis, where you can imagine that one of these mobile elements might hop into an important gene in the coding region and disrupt its function, or it might insert in a regulatory sequence and affect gene expression. And so this is the basis, we believe, for a number of diseases. And so we think that transposons are linked to cancer and also the process of aging. Other diseases are listed here that we believe are associated with transposon mutagenesis. So we've got a type of muscular dystrophy, we've got 2 types of hemophilia, A and B, and that's when you lack a clotting factor. And we have a type of severe immunodeficiency as well, all believed linked with transposon-generated mutation. So pseudogenes, so they have sequences that are very similar to known functioning genes, but they are non-functional. So we consider them to be historic relatives of functioning genes that can't express RNA anymore. And they're usually duplicate or disable copies of that original coding gene. So they usually have strong homology to the gene that they originated from, and they're, by definition, they are non-functional, at least in the way that they were originally intended, as they don't encode a protein anymore. So we believe there's up to 25,000 human pseudogenes in our genome. And in the past, they were categorized as junk DNA, but we've learned a lot more about them and believe they actually are important in terms of gene regulation and gene expression. And one important consideration is that during genetic testing, we have to factor in pseudogenes because these very similar sequences potentially can create confusion for us. And also over here, I put in a diagram from an article entitled Human Line Retrotransposons Generate Processed Pseudogenes. So there's two mechanisms for the creation of pseudogenes. So there's a duplication and mutation mechanism to give us duplicated pseudogenes. And over here, we have a different method which creates what we call processed pseudogenes, whereby you get an RNA transcript and it gets processed, and then reverse transcriptase converts it back into DNA, and then it can insert back into the genome. And so the creation of pseudogenes, as I mentioned earlier, is linked to these different mobile genetic elements. And also they're relevant because we believe they're involved in the pathogenesis of a number of cancers. So what you can see here in this table is that pretty much every category of DNA that we talked about is associated with a certain disease or disorder. And we've got some examples listed here, and they're categorized into DNA of known function and unknown function, and then further broken down into, are they single copies, multiple copies, or non-coding. And of course, down here, we have our pseudogenes, and because the process whereby you create a disease-causing gene that can be called gene conversion. Then we have the centromeres, additional diseases, rheumatoid arthritis, for example, lupus, a chronic inflammatory autoimmune disease, hemophilia, transposons, and then the mini and microsatellites. And we have a number of disorders here that can be associated with this. And up here, of course, typically what happens if you disrupt the protein coding part of a gene, a lot of disorders involve deficiencies of a particular enzyme in a metabolic pathway. So we have phenylketonuria, or we have changes in protein structure, and that's underlying basis for sickle cell anemia and beta thalassemia. You can have changes inside the gene, or you can have changes in the regulatory sequences or where the RNA gets processed as well, and these can all cause changes in the structure of protein. Down here, we have microRNAs. MicroRNAs are believed to have a role in controlling gene expression. So we have some diseases associated with that as well. And then we have a collection associated with what we call multiple copy sequences and a range of diseases here. So this is here is the central dogma in molecular biology, whereby information is transferred in the cell from our blueprint or in DNA, of course. All the information about our cells is encoded on genes in DNA. And when DNA is expressed, it's first transcribed into messenger RNA, and then RNA is translated into protein, and protein does the various jobs in the cell. In addition, we replicate DNA as a normal process, and we've talked about the involvement of reverse transcription, for example, with transposons and pseudogenes. RNA replication isn't something that occurs in humans, but we do know, for example, some viruses, they have RNA in their genomes, and they have to replicate that. So Obviously, then we need to be able to distinguish structurally in the forms of RNA and DNA as shown here. Both DNA and RNA, of course, are made of nucleotides, and the basic structure of a nucleotide is a base, a nitrogenous base, a sugar, and a phosphate group. The four bases in DNA are A, G, C, T, or adenine, guanine, cytosine, and thymine. And they're most of the same in RNA, except we replace thymine with the base uracil. And we can also categorize the bases in terms of pyrimidines and purines. And you can see that pyrimidines are derived from a single ring structure and purines have a double ring structure. So cytosine, thymine, and uracil are all example of pyrimidine bases, and adenine and guanine are example of purine bases. So when nucleotides come together and form polymers, that's nucleic acids. And what happens is the 3' hydroxyl group on a sugar in one nucleotide binds to the phosphate group on the 5' carbon atom in the sugar of the next nucleotide. And so here we have a sequence of RNA. I know it's RNA because it's got a uracil. And also if I look at the ribose sugar, it has a oxygen here. So I know that it's RNA. And this is the free 5' end. And we always read DNA from the 5 to the 3' end because that's the sense strand or coding strand in both DNA and RNA. And here we can see this free phosphate group at what we call the 5' end. It's called 5' because it's at carbon number 5 on the ribose sugar here. Okay. And then we have a OH group at carbon number 2. Okay. So what happens, we have a chemical reaction between the phosphate group on one part of the sugar and with the 5' carbon atom on the next nucleotide. So we call the sequence the primary structure when we talk about the sequence of DNA and RNA. So we have the 5' unreacted phosphate on that end, and we have the sugar on the other end with the free 3' hydroxyl group. So that's the free 3' end. So what happens is when nucleotides join together, they form a phosphodiester bond. So again, once more, we have the nucleotide here. Here's the hydroxyl group at the 3' position on the sugar. And here we have another nucleotide, and here's the phosphate group. So we get an ester bond as a reaction between these two groups here, and we get the formation of a 3' to 5' phosphodiester bond. So this is the reason why DNA is only synthesized in the 5' to 3' direction. We have to add the nucleotides in this order so that these groups can react to form phosphodiester bonds as shown here. So now we have a short piece of nucleic acid, and once again there's the free 5' end with the phosphate, and then we have the free 3' end with that hydroxyl group. So looking at the structure of DNA in a bit more detail, so DNA stands for deoxyribonucleic acid, and it has a deoxyribose sugar, and it's called that because it doesn't have an oxygen at this position. There is no oxygen atom on carbon number 2 in deoxyribonucleic acid. It's a double-stranded molecule or helical structure. This was first discovered by the scientists Watson and Crick, and they won a Nobel Prize for their work. They used a combination of X-ray diffraction and other methods, to work out this double helical structure. And if you liken it to a ladder or a twisted ladder, the backbone of it is made of sugar phosphates. And the rungs of the ladder are made of the bases, the 4 different bases. And you can see there that they bind to one another in a characteristic sequence. So what we see is that G's only base pair with C's and A's only base pair with T's. And what holds them together is hydrogen bonding. And the two strands are anti-parallel. That is, they run in opposite directions. So you can see that this strand starts at the 5' end and runs on through to the 3' end. And the other strand runs in the opposite direction. So they are anti-parallel strands. And here we have a little bit of pharmacology. We have a couple of anti-cancer drugs used to treat leukemia. And the way that these two drugs work, they insert between the bases of the DNA and they interfere with the activity of an enzyme called topoisomerase 2, which we're going to talk more about in a minute. But it's an enzyme that's required for the proper replication of DNA. And then we've got an example of another drug. This also binds to the DNA structure and it actually distorts the structure of DNA and we get malfunction as a result. So there's some chemotherapeutic drugs that work by interacting with the structure of DNA. We need to be able to compare DNA with RNA, and this is a very handy diagram for doing that. RNA, of course, stands for ribonucleic acid. It's single-stranded, whereas DNA is double-stranded. It has a sugar phosphate backbone the same as DNA and has the bases in the middle. And it has a different structure here with the ribose sugar, which has an oxygen at the 2' position, which we believe gives extra stability to the molecule. So when cells divide, they undergo mitosis. They have to overcome certain challenges in DNA replication. So remember that DNA is double-stranded, and we before we can replicate it, we actually have to separate these strands out and then copy them. And we form these complementary copies. We form these new DNA strands using the original strand as a template. And we form what we call daughter DNA strands. So we have to be able to unwind and separate those two DNA strands. DNA is very tightly wound and compacted in the form of chromatin. And when we do unwind it and get single-stranded DNA, we have to protect that DNA from enzymes called nucleases, which will attempt to break it down. And we always synthesize new DNA from 5' to 3' end. And what we have here, of course, is template strands in opposite directions. We have one that's 5' to 3' and one that's in the opposite direction, 3' to 5'. And lastly, we have to guard against errors in replication by making sure the correct base is added. And if you don't fix these errors or prevent these errors, then the consequences can be catastrophic. So the DNA unwinds at a specific point. We call it the origin of replication. And in higher organisms, we have multiple origins of replication where you're getting bidirectional replication. So the replication is moving in opposite directions in each one of these origin of replications. And we call them replication forks, are the points at which new DNA chains are being formed. And you can see as the replication continues, these bulges get larger and larger as we form copies or duplex daughter DNAs. And at the end of this process, we now have a complete identical copy of the original DNA strand. So if we look at the eukaryotic replication fork up close, a bit more detail here, you can see there's a assortment of enzymes and proteins which are carrying out the process of replication. And here we have the original DNA strand, and it's been separated into two single-stranded DNA templates, and we have a DNA polymerase going along and reading the sequence and making a new synthesized daughter strand. This strand here, the top strand, is called the leading strand template, and down the bottom we have the lagging strand. And so process is different in DNA replication between the two strands. So an overview of this process: firstly, obviously we have to unwind the strands, and that's done by an enzyme called helicase, which is a helix destabilizing protein. And to protect the single-stranded DNA from being broken down by nucleases, we have a single-stranded binding protein called RPA, which binds to it. And then we have something called primase activity. So initiation of replication of DNA requires a RNA primer, which you can see here. And this has to be created by a primase enzyme. And this activity is actually associated with a type of DNA polymerase called alpha. Once we incorporate a few nucleotides, then we get the second DNA polymerase coming along, delta, and it's associated with a couple of proteins. So you can see it here, and also it's on both the leading strand and lagging strand. So you can see it's associated with a couple of other proteins, PCNA, proliferating cell nuclear antigen, and also replicating factor C or RFC. And they enable the polymerase to bind to the template and do its job. Also, we have these two enzymes, FEN1, shown here, and RNase H1. Their job is to break down these RNA primers we had at the beginning. Once we're finished with them, we need to break them down and remove them. And lastly, we need to seal the gaps in the DNA between the fragments, and DNA ligase comes in at the end and joins the DNA pieces together. So if we look at that, the way the two strands are replicated in more detail. So this is a model called the semi-discontinuous model for DNA replication. So once again, there's our original strands, and we separate them here into the two single strands, which are used as DNA templates. And again, DNA polymerase can only make DNA in the 5' to 3' direction. We have to do the different strands in a different way. So the leading strand is pretty straightforward. Because the template is in the 3' to 5' direction, it is synthesized continuously. The new strand is made from the 5' to 3' end in one continuous piece, as shown here, reading its complementary strand, the template 3' to 5'. Lagging strand is different because it's in the opposite direction, and this one, the template reads from 5' to 3'. So in this case, we have to synthesize the complementary DNA in short bursts, producing these fragments called Okazaki fragments. And at the end of this process, we then join these fragments together with the enzyme DNA ligase. So DNA polymerase, its job is to add nucleotides sequentially to the growing DNA chain, and again, in direction of 5' to 3' only, because remember, we have to form these phosphodiester bonds, and we do it by bringing a nucleotide in with its phosphate group at the 5' end, which then interacts with the 3' hydroxyl group as shown here. And that's basically what the enzyme does. It brings the nucleotides together, and it allows this chemical reaction to occur. So we always have the last nucleotide added with this free 3' hydroxyl group. And what happens that this is a nucleophile, and there's a chemical attack on the phosphorus on the incoming nucleotide. So the DNA polymerase brings the next nucleotide in, and it has the phosphate group here, triphosphate on the sugar. And we get the elimination of phosphate and the formation of a new phosphodiester bond as a result. RNA polymerase, of course, cannot produce DNA from nothing. We need certain ingredients for this process to occur. So as we mentioned earlier, you've got to have an RNA primer and a short sequence of RNA. It binds to the template DNA, and then we make the new strand from that at the beginning. You need the building blocks of the new DNA that you're going to make in the form of deoxyribonucleoside triphosphates, DTTP, DATP, D-GDP, and D-CTP. You need the cofactor magnesium. Obviously, you need the DNA template that the RNA polymerase is going to read. And also you need the components of the RNA primer in the form of ribonucleoside triphosphates. So there's actually a whole bunch of different eukaryotic DNA polymerases. We're going to focus on just a few of them. There's at least 19, and 5 have been characterized in more detail, as shown in this table. And you can see the table is comparing them on the basis of their size, any associated function. We have something here called exonuclease, which is involved in proofreading. The primase we mentioned, making the RNA primer, and you can see that only the alpha form can do that. And also their efficiency. So some are faster than others, call that processivity. And also we have fidelity, and high fidelity means low error rate. And also other roles such as repair are summarized in this table here. So ones we'll focus on, the alpha polymerase again makes the primers. The delta polymerase is the principal DNA polymerase in eukaryotes that makes most of the DNA, and we require that pCNA protein for it to bind to the template. And we have polymerase epsilon, and has a number of roles. So it's also involved in leading strand replication. It's involved in proofreading because it has an exonuclease activity. And we believe it may also replace this polymerase on the lagging strand as well. So what is proofreading? Well, proofreading means removing and replacing incorrect nucleotides during the process of DNA replication. And proofreading improves fidelity greatly, reduces the error rate considerably. So it improves it from 1 in 10 to the 4 to 10 to the 5 base pairs to 1 error every 1 in 10 to the 9 to 10 to the 10 base pairs. So that is a big difference. So the polymerases that are associated with proofreading are the delta and epsilon types of polymerases. So what occurs is occasionally a polymerase will make a mistake. It will incorporate the incorrect nucleotide in the new strand of DNA. And as a result, you get a mismatched pair in many cases, because remember that only certain bases bind to other bases. G only binds to C and T only binds to A. And where we have a region of mismatch, that's a structural abnormality that is detected by the polymerase. And then the polymerase can use the 3 to 5 prime exonuclease activity to remove the incorrect nucleotide, and that is then replaced with the correct nucleotide, and the process of DNA synthesis can continue. The pCNA that we mentioned earlier is a beta sliding clamp, and this enables the polymerase to bind to its DNA template, and it's made of a trimer of 3 identical proteins that surround the DNA, and there's 2 different views of it. So we have a ribbon structure, and over here we have a 3-dimensional molecular model with the DNA in the middle. And at the very beginning of the process, shown over here, on the right-hand side, we have topoisomerases. There's 2 different types. And what's its job? Well, it's there to relieve the strain of unwinding of DNA due to supercoiling. So DNA is highly supercoiled, and we need to be able to relax these structures. And these enzymes do it by causing a break in the DNA. And the type 1 topoisomerase is abbreviated TOP1. They cause a single-stranded break in the DNA strand, and whereas the type 2 cause a double-stranded break. They cut both strands of DNA helix to relax the supercoils, and these type require ATP, whereas type 1 don't. Here's a useful summary flowchart from Lippincott. You might find this useful for your revision because it summarizes the key points of both DNA structure and DNA replication. So here again, it's reminding us that we've got a phosphodiester backbone, deoxyribose sugar, and DNA, and we have the 4 bases listed there. And it is a double-stranded helix with anti-parallel strands. and we read the sequence always from the 5 to the 3' end. It's held together with hydrogen bonding and other interactions between the bases. And A always binds to T and C always base pairs to G. When we replicate DNA, we have to make it single-stranded. We have to unravel that process. And that process begins at the origin of replication. And we create our replication forks. They're bidirectional. We have to separate the strands using helicase, and we control the supercoiling using DNA topoisomerases. DNA polymerases bind, make the new DNA strands, and this proceeds in the 5 to 3' direction. And we require an RNA primer for that. And we are copying both strands of double helix, and we make 2 daughter strands. We have a leading strand, lagging strand, which is synthesized differently. Leading strand is synthesized continuously in the 5 to 3' direction. We only need 1 RNA primer for that. Whereas the lagging strand is made discontinuously in the Okazaki fragments in the same direction, but away from the fork. And for this, we actually need many RNA primers. So useful summary for you. Of course, it's more complicated than that because we have to factor in the cell cycle. So we have multiple origins of replication in eukaryotes. It's much simpler in bacteria. We need to control the timing relative to cell division, and therefore this involves more proteins and enzymes. And the cell cycle is where cell growth and division are divided into different phases, M, G1, S, and G2. So what are these different phases? Okay, so M phase is mitosis, where you get the physical separation of the cells into daughter cells. G1, gap phase, is the longest part of the cell cycle. We have rapid growth and metabolic activity, the cells preparing themselves for division. Sometimes those cells can go into GO phase, a dormant phase, and this is something we see with nerve cells. S phase is the time of DNA synthesis, replication, and growth, and that is followed by G2, which is a relatively short period, and the cell is preparing for division. And these 3 phases here collectively are called interphase, by the way. And then we have, of course, mitosis. So cell cycle times do vary a lot. Some, when you have rapid turnover cells, such as those lining the digestive tract, the cell cycle time be less than 24 hours, whereas others can have times up to hundreds of days. So replication of DNA can only be initiated by chromosomes from cells that have reached G1. And in step 1, we have a variety of proteins. We have a origin recognition complex, or ORC, which binds to the DNA all the way through cell cycle and enables other proteins to attach to control replication, such as RAP, replication activator protein, and that prepares for the start of DNA replication. And thirdly, we have replication licensing factors, proteins that are essential for DNA replication, and many of them are cytosolic. And they all come together to form a pre-replication complex, as shown here. And this makes the DNA ready for replication. In the next stage, step 2, requires other proteins and protein kinases. These protein kinases are called cyclin-dependent kinases. And what they do, they bind to cyclins. What cyclins are? They're transient proteins produced in one part of the cell cycle and then broken down in another. And what they do is they combine with these CDKs and they activate them. And what do kinases do? Well, they phosphorylate things. So they activate or initiate DNA replication by phosphorylating a couple of these groups of proteins from the pre-replication complex. So you can see we've phosphorylated the RAP and the RLFs. And this triggers the process of and commits us to replication of DNA, which is going ahead here in S phase. After this this process is initiated, the RAPs and RFs, they get degraded, and this enables a transition through G1 to S phase. So once we've reached the G2 phase, if everything's gone well, DNA will have been replicated. And then we move on to mitosis. In mitosis, we get the separation, the equal separation of DNA into the daughter cells. And at the same time, we get the entrance of the replication licensing factors, because during mitosis, the nuclear membrane dissolves, and the RLFs, which are made in the cytosol, are able to enter into the nucleus. And this means that the daughter cells can then initiate a new round of replication. Telomeres are special structures, or caps, we find at the ends of eukaryotic chromosomes, as shown here, and they protect the chromosomes when cells divide. And what we see over time is telomeres, they get shorter and shorter, and therefore they're an indication of the general health and the age of an individual. So they are something that's associated with the process of aging. They consist of a series of repeated DNA sequences, TG-rich repeats, and they're non-coding, and they are a buffer against the breakdown of DNA sequences at the ends of chromosomes during replication. So there is an enzyme called telomerase that's associated with telomeres. So it's a ribonuclear protein. It contains a piece of RNA that's complementary to part of the telomere. So what telomerase do, it binds to the 3' end of the telomere sequence along with its RNA template, and it produces copies of the DNA by addition of bases and restores the telomere back to its original length, and this process is finished by DNA polymerase. So it's a mechanism for synthesis of telomeres, but you only see this enzyme normally active in, for example, embryonic cells, also germline cells and stem cells. It's not normally active in adult somatic tissue. If it is active, then it's pathological. So, for example, we see this enzyme telomerase is reactivated in cancer cells, and therefore it is a target for chemotherapy and also development of new drugs against cancer. So finally, we have this diagram, inhibitors of eukaryotic DNA replication. So here we have, once again, the cell cycle, and you can see that we can target the cell cycle with various groups of drugs. And this group of drugs here are all targeting S phase. So when we treat cancer, we're generally targeting some aspect of the cell cycle. So some examples of drugs, we've got cytosine arabinoside, and here it is down here. It's also called cytarabine, and it's an example of a pyrimidine analog. So you remember that you have the two major groups of bases in DNA, purines and pyrimidines. So this is an analog for a pyrimidine, and it inhibits the normal process of DNA replication, preventing chain elongation. And we have another drug here. This is actually a purine analog, and it works as an antiviral drug and also inhibits DNA chain elongation. This drug here, actinomycin, also known as actinomycin D, this inserts into the grooves of the DNA duplex, interferes with both DNA replication, but also gene expression, RNA synthesis. That's another example of a anti-cancer drug. So you can see here that collectively we call all these different groups of drugs are referred to as antimetabolites. So they're drugs that interfere with normal cell processes, and all of these here are targeting DNA synthesis that would normally occur during S phase. Another good example is methotrexate, and this is known as a folate analog. So folic acid is an important component of nucleotides, and that's how methotrexate works. So if you have any further questions, you are free to email me, and I thank you for your time.